The overall goal of this procedure is to create, test, and validate genetically encoded inducible artificial transcription factors, or ATFs, with desired specificity. This is accomplished by first creating the ATF via a simple yeast transformation in which inclusion of a DNA binding domain PCR product results in an in-frame fusion with a human estrogen receptor and VP16. The second step of the procedure is to create a GFP reporter plasmid for the ATF by replacing the binding sites in the plasmid PMN8 with ATF targeting sequences. The third step of the procedure is to assay the ATF's ability to activate the GFP reporter and to also assess its effect on yeast physiology by measuring the growth rate. The final step of the procedure is to use the validated ATF to conditionally express a native genomic target. Ultimately, any target gene of interest can be made conditionally active.
The main advantage of this technique over existing methods like galactose-mediated induction of gene expression is that selective induction can be achieved under diverse nutrient and physiological conditions without pleiotropic effects.
This method can help answer key questions in the yeast biology through the fast and reversible introduction of desired gene products, enabling study of both gain and loss of function.
To begin, PCR amplify the DNA binding domain, or DBD, of interest using a high-fidelity polymerase. Transform greater than 1 microgram of the purified PCR product into yeast using the lithium acetate method. Following transformation, spin the cells at top speed for 15 seconds in a microcentrifuge. Remove the transformation mix and resuspend the cells in approximately 200 microliters of